So hello everyone. This is a short video, very short video on neoplasia of the cervix. So I would like to go on a very quick account, and I would like to teach you in the best way possible what is neoplasia, what is the neoplasia of the cervix, and what is cervix. Actually, we have something which is known as an ectocervix and an endocervix. So if you just remember the female uterine tract. So if this is your uterus, the upper portion over here, this is your uterus, then comes the endocervix, then comes the ectocervix, and here it continues as the vagina in this manner. Okay, so this is the vagina. Now if I draw the lining epithelium, so the endocervix, it's actually columnar epithelium. So you have beautiful columnar cells in the the endocervix. Yes, the columnar epithelium continues as an endocervix, as an ectocervix. Endocervix and the ectocervix consist of the columnar epithelium. In the case of, the premenopausal, or the child of age 5, 6, 7, 8, 9, 10, 11, at this particular ages, the ectocervix is still the columnar. But what happens gradually is, because of the production of the estrogen, during every cycle, estrogen is produced by the thicker cells of the uh, ova, and that thicker cells of, actually I should say, thicker cells of the Graafian follicle, that would be much more precised. Moreover, the thicker interna cells of the Graafian follicle. And when that estrogen is continuously produced, that estrogen is responsible for conversion of the ectocervical columnar epithelium into the stratified squamous. Okay. So now, if this is your ectocervix, now here this particular will not be there. Here it will be, initially it will be, of course, the endocervix will be columnar, and then here it will be your squamous. Okay, so this will be your stratified squamous. So, and the area and the area uh, where the ectocervix and the endocervix is, just where the part of the columnar epithelium ends, and when there is a beginning of the stratified squamous, that area is known as a transformation zone. Okay, and that transformation zone is highly prone to a virus which is known as HPV, okay, human papilloma virus, HPV. And this HPV is responsible for causing your neoplasm of the cervix. One more thing I would like to add here is the HPV, it consists of certain component like the there are different types. Actually, there are different types like type 8, type 10, type 16, 18. But the most important is type 16 and 18. Type 16 and 18 are the most important. Type 16 and type 18. They are the most important component. Why they are the most important component is they're responsible for they liberate out certain proteins. Okay, these proteins actually, they're basically the E7 and the E8, okay, respectively. E7 and the E8. So type 16 and 18, they're responsible for the production of proteins like E7 and E8. And these E7 and E8 are responsible for the inactivation of the P53 and retinoblastoma and RB. Okay, retinoblastoma. I'm concising it in the best way possible. This will be really easy for you guys. Okay, so once again, whatever I talked till now, there are two areas. One is the endocervix, the other one is the ectocervix. The endocervix is lined by the columnar epithelium, and the ectocervix is lined by the stratified squamous. The area of ending of the endocervix, that is basically lined by the columnar, and the area of starting of the ectocervix, which is basically lined by the stratified squamous later in life. Okay, that area, this junction is known as the transformation zone, which is highly prone to human papilloma virus. Now straight away, let's go on to the topic proper. Uh, so type 16 and 18 are the most important, and these type 16 and 18 are responsible for secreting proteins like E7 and E8, and these proteins are responsible for the inactivation of the retinoblastoma and the P53. Okay, actually type 16 is for P53 and type 18 is for retinoblastoma. Okay, so and remember, P53 and retinoblastoma, they are tumor suppressor genes. Now, what are tumor suppressor genes? This component of your neoplasia. I just like to recap a little bit. Now, the cell cycle which is continuously going on, it's basically uh, that cell cycle continues because of the activation of the cyclins. Okay, that cell cycle continues because of the activation of the cyclins. So the cell cycle has been continuously, operating. There are certain checkpoints, and these checkpoints are again looked after by certain tumor suppressor genes, which doesn't let the neoplastic cell to go under the cell cycle. And when these tumor suppressor genes are basically mutated, which is basically the P53 and retinoblastoma, they're basically the tumor suppressor genes. And when these tumor suppressor genes are, they're innervated, they're inactivated or mutated, then there is proliferation of the cell that will lead to neoplastic transformation. Now, let's go on to the um, uh, let's go on to the CIN, cervical intraepithelial neoplasia. Okay, so for that, I would erase everything on the slide here, and then uh, okay, so now I would like to talk about, whenever there is a question about, okay, classify cervical intraepithelial neoplasia. What is the classification? Then you have to say it's SIN 1, SIN 2, SIN 3. Cervical intraepithelial neoplasia 1, cervical intraepithelial neoplasia 2, and cervical intraepithelial neoplasia 3. Now, you have basically SIN 1, SIN 2, and SIN 3 respectively. 2, and then you have 1, 2, 3. Now, what you need to remember here is, which is very, very important aspect, I just took time to make this wavy pattern, and you might be slightly in confusion that why am I making, actually the stratified squamous epithelium doesn't look like this for sure. Big, but for your understanding, for my easiness to make you guys understand, I'm making it in this pattern. Okay, so I hope you there are three layers that I've been making. The one, two, and three. Okay. So I will be dividing this cervical epithelium. Basically, I'm talking about the transformation zone. Okay. Transformation zone. Now I'll be dividing this transformation zone into three aspects. Three aspects. So this is area number one. This is area number two. This is area number three. Okay. Now, if the neoplastic cell is only involved in this lower one third area, lower one third. So it's one, two, three, right? So if the neoplastic cells are only involving the lower one third, it's known as SIN 1. Okay, next. If the neoplastic cells are involving the lower two-third, that is this part and this part, lower two-third, it's known as SIN 2. And if the neoplastic cells are involving all the layer, then this is known as SIN 3. And remember, SIN 3 it does have a high progression to convert into the squamous intraepithelial neoplasia. Okay, and now you might be confused, like what is we yes, we are talking about squamous intraepithelial neoplasia. So why are you saying that high grade of conversion to squamous? Actually, I meant to say that invasive squamous intraepithelial neoplasia. Okay, so what is the invasive component and what is a non-invasive component? I would like to make you clear about this. See, invasive component means I've made this particular barrier, isn't it? I've made this particular barrier over here uh, at the sector. This barrier demonstrates that the epithelial transformation into neoplastic, okay, neoplastic transformation, it's above the basement membrane. It's above the basement membrane. It has not breached the basement membrane. It has not breached the basement membrane. I would like to show you in the Robbins picture. Wait a minute. Okay, so I just took my time to show you this particular diagram from the Robbins anode. Okay. Now, see the normal epithelium, it's so clear. The stratified squamous epithelium, it's so clear, it's so beautifully illustrated in this diagram. And you can see these dysplastic changes of the epithelium in the lower one third. Okay. And now, and if you see in scene 2, the dysplastic changes, it has occurred in this area, and there are some more dysplastic changes at this area. The above one, they're clear of dysplastic cells. There are no dysplastic cells. You can clearly tally these cells with these cells. They are somewhat similar. Okay, a central nucleus with clear cytoplasm. This nucleus and this nucleus, it's kind of looking similar, and the same similarity is seen here also at the top. But if you see in the scene 3, then you can see the dysplastic changes is occurring in all the level of the epidermis, all the level. Okay, now I meant to say that this is the basement membrane over here. This is the basement membrane. The tumor cell has not breached the basement membrane. So basically, whenever the tumor cell breaches the basement membrane, you're going to have desmoplastic reactions that is shown basically by the stromal cells. Okay, and that desmoplastic reaction tends to make the cervix, more firm in texture, and you would have a barrel cervix, which is a critical finding of the invasive type of cervical intraepithelial neoplasia. Barrel cervix means like a barrel, okay, like this one, curved barrel. Now, I would again want to pause. So clearly we can tally that picture with this one. So now, how would you write your histology? How would you talk about your histology? Wherever you will be talking about the, okay, cervical intraepithelial neoplasia, type 1, type 2, type 3. Type 1 involves the dysplastic cells in the lower one third. Now you might be asking, what about the above uh, above area? I showed you, if you remember, I showed you the stratified squamous epithelium will not look, like the previous one. I showed you a picture where there was a cell, and inside there was nucleus in this way. And I showed you the same pattern was seen in scene 1 and scene 2. I showed you, right? There was a clear cytoplasm. Yes. And what is that? That is known as coilocytic atypia. Once again, I would like to type here coilocytic atypia. Atypia. So coilocytic atypia, it's basically seen in scene 1 and scene 2, but it's not seen in scene 3. That's the differential. So if you find dysplastic changes in the lower two-third, and if you find coilocytic atypia at the above area, at the above upper one-third, and if you don't see any kind of breaching, the basement membrane, any breaches in the basement membrane, then that's a critical case of sin 2. I won't say critical because, sin 2, it's not as bad prognostic as sin 1. Sin 1, it's a good prognosis. It's not a bad prognosis. But the worst prognosis here would be sin 3, if this is clear. Now, there's some new type of, there's a new classification which says that the low grade squamous intraepithelial lesion is scene 1. The low grade, okay, the low grade is scene 1. And if you talk about the high grade, it's basically scene 2 and scene 3. Okay, and out of scene 2 and scene 3, the most worst prognosis again is scene 3. I think that was clear. This was all about cervical intraepithelial Neoplasia. We did not talk about the invasive component a lot in this particular sector, but the invasive component is also somewhat similar. Thank you. If you like this video, subscribe to my channel. Good luck.